All right, we're going to get into the second part of DNA structure. We're going to be getting into or looking at RNA, what RNA is, how RNA is formed. Then we'll get into DNA replication at the very end and look at how DNA and RNA are both related in that process. So let's go ahead and get into this. We know RNA it's considered or known as the helper molecule. It stands for ribonucleic acid. The sugar that makes up RNA is called ribose, a little bit different than the deoxyribose sugar that makes up DNA. Remember, this is also RNA is also a polymer of a nucleic acid. So the nucleotide is still made up of nucleotides. That would be the monomer. Some of the functions of RNA. The big function is it's kind of like the say the DNA is the blueprint. Okay, if you're looking at blueprints, and the DNA would be the blueprint of how everything is supposed to be built. Well, it's kind of hard to understand that blueprint, so the RNA's job is to be able to decode that blueprint or to understand what the blueprint means and write the message in terms that the organism can understand. So the main function of RNA is actually to carry the messages from the DNA out of the nucleus into the cell, and then those messages are then going to tell the cell how to make protein. There are three different types of RNA. Messenger RNA, the name implies it's the type of RNA that carries the message from the DNA in the nucleus out into the cell. The transfer RNA is going to transfer things called, and we'll get into this next unit, we're going to transfer things called the amino acids that build the protein. It's going to transfer them to the ribosome, and then the ribosomal RNA is going to be the type of RNA found in the ribosome that helps build the protein. Okay, so RNA is still made up of nucleotides, very similar to DNA nucleotide. It has a 5-carbon sugar, but the sugar is ribose. It also has a phosphate group, okay, and the sugar and the phosphate make up that rung or that backbone, and then it does have a nitrogen base. So there's your sugar, there's your phosphate, and then there's your nitrogen base. Very similar to the nucleotide of DNA. If we were to label the parts of just an RNA strand, we know that would be your sugar, there would be your phosphate, and then there would be your nitrogen base. Notice the picture is different than DNA because remember on DNA there would be another strand up here in the picture. So this is only single-stranded. You notice there's a nucleotide, here's a nucleotide, here's a nucleotide, and so forth. It's just a single strand of nucleotides, whereas DNA is a double strand. All right, let's look at some common differences between DNA and RNA. We know DNA has 2 strands, it's double-stranded. RNA is single-stranded. Now that's important because it takes less energy to make. It makes it smaller so it can actually leave the nucleus. It can travel from the nucleus through the nuclear pores out into the cell to carry the information. The sugar, deoxyribose versus ribose. And the base pairing is just slightly different. It's almost exact same, but it's slightly different. So let's look at the base pairing for RNA. You still have cytosine and guanine pairing together through a triple bond. But adenine is going to bond with uracil, not thymine. Thymine is not used in RNA. A uracil is going to replace thymine. It's a smaller, more simplified version, if you will. It can still bond with adenine, but it makes it to where the RNA can be single-stranded. It makes it less complex. So for our purposes, that's enough. We know that RNA is still going to have cytosine and guanine and adenine, but it's going to use uracil instead of thymine. So uracil is going to take the place of thymine for RNA. Okay, so here's how it works. You have a double strand of DNA. Okay, it gets to make RNA. The DNA is going to be unwound. It's going to be untwisted, so it to make it to where it's more functional. You have the left side of DNA strand, you have the right side, okay? What's going to happen is you're going to have an enzyme come along. Well, let's look at the parts first. You have cytosine, remember it bonds with guanine. You have adenine with thymine and so forth. Okay, so there's our DNA strand. What's going to happen is an enzyme is going to come along called DNA polymerase, and it's going to unzip the strand of DNA in a certain section because the RNA is going to be smaller. It's not going to be as long, okay, and it's going to be single-stranded, so you don't have to unzip the whole DNA strand to make a strand of RNA. You can unzip a smaller section. So it's going to the DNA polymerase is going to come along, that enzyme, and unzip, which means it's going to break those hydrogen bonds holding those nucleotides together. It's going to unzip them. Once it does that, then another enzyme called RNA polymerase is going to come along, and it's going to build the complementary RNA strand. Okay, so if that's cytosine, then it'll attach a guanine nucleotide. An adenine for DNA is going to attach a uracil nucleotide. Guanine for cytosine, and then a thymine nucleotide is going to an RNA strand that be complementary would be adenine. Okay, so a quick preview of DNA and RNA. We know the deoxyribose is for DNA, ribose is for RNA, adenine is present in both, cytosine is present in both, guanine is present in both, thymine is only found in DNA, uracil only found in RNA, and a double helix means it's double-stranded is only going to be DNA. RNA is single-stranded. They're both going to be made in the nucleus, okay? The RNA can leave the nucleus and travel to the cytoplasm. DNA is going to store the genetic information. RNA is going to interpret that genetic information. Okay, last but not least, let's get into DNA replication. We just looked at how RNA is made. RNA is going to be made, and we're going to get into what its specific purpose is later. But DNA always needs to be replicated. Our cells wear out. Okay, they wear out over time, and new cells have to replace them. Before new cells can replace them, you got to make sure that you have an extra copy of the DNA to put into that new cell. So this is where DNA replication comes into play. During replication, you start out with your DNA strand, okay? Your DNA strand, before the cells replicate, before new cells are made, the DNA strand is going to be unzipped by DNA polymerase enzyme. Then you have nucleotides that are going to come along and be used by that DNA polymerase enzyme, and it's going to build complementary strands. Once it does that, you have two identical sets of DNA. One goes to the original cell and one goes to the new cell that's going to be made. So here's the process. All right, step one, you've got your strand of DNA. The whole strand of DNA has to be unzipped by DNA polymerase. Once it does that, you have one side and another side. You have two separate pieces. Then DNA polymerase is going to come along and build a complementary strand to both pieces of the DNA. Once it's all done with the whole set of DNA for that cell, then you have not one but two sets of DNA. One will stay with the old cell, and then one set will go to the new cell to help when it needs to be used to be replaced.